So I'd like to thank Terry and all of the people at MICH for the opportunity to present some of their research. And while I sent this title some time ago to NG to kind of some of the work that we're doing using the xenograft model to study human T-Lymphoblastic leukemia, I also wanted to take part of the time to talk a little bit about what we're doing in the field of regenerative medicine for another condition. And that way, give you an idea of the spectrum of what I'm doing, what our lab does with respect to stem cell biology as well as transplant and regenerative medicine. So, first of all, I'd like to thank the people who actually did this work and our collaborators who supported this work. I had the good fortune of doing my postdoc with John Dick at the University Health Network, as well as having collaborators who kindly biobanked the leukemia specimens, and certainly the funding agencies that supported this work. So as many of you know, acute leukemias are blood cancers, and certainly in children this is the most common cancer of childhood. It's less frequent in adults, but actually more lethal. And so while there are commonalities with respect to classifications, certainly treatments and survival are significantly different between kids and adults. And of course, we classify leukemias based on their blood lineage, and we use the human hematopoietic map to label our leukemias based on the lineage. So I studied the lymphocyte lineage in normal development, and so in the neoplastic side, I studied the lymphoid leukemias, and certainly in terms of the lymphoid lineage, I focus on the T lineage, acute T-Lymphoblastic leukemia. So, as many of you know, childhood leukemia has had a really good outlook because increasingly aggressive treatment protocols have really improved survival. The survival in childhood ALL across the board is extremely high, with 80%, 85% survival in adults, not as good. Now, even though these protocols have improved survival, these are not without cost. So if you look at the long-term morbidity or mortality for leukemia patients, over time, even if they do not have relapsed disease, there is a significant mortality long after they have been disease-free. And certainly with certain protocols, there's a price to be paid with toxicity related to side effects of the chemo drug or development of secondary neoplasms. So it's not enough that we just nuke the leukemia. We have to look at what the long-term morbidities are like. And certainly in adults, there's even more documented morbidity and mortality, probably because some of the cytotoxic treatments induce such severe myocardial or other myelosuppressive consequence that they don't recover. And certainly, we have to recognize there's still treatment failure. There are still a cohort of patients that just don't respond to treatment, and salvage is the only thing, the only salvage treatment that we can offer is bone marrow transplant. So, and I'm sure you all know bone marrow transplant survival is not fantastic, especially in kids. So clearly there is room for improvement, and if there are clear goals to achieve in terms of improvement in TAL outcomes, it is to limit the toxicity and to predict those who won't respond to primary treatment or are going to relapse early, and give them either salvage treatment early or experimental treatment sooner, because time is clearly of the essence for relapse patients. So clearly there is a need for us to be able to some way identify who are the patients that are not going to respond, who are going to relapse, and give them secondary therapy sooner. And frankly, for TAL, there is no way. We don't have a classification system or prognostic marker that's sensitive yet and validated yet. So in my lab, we've worked on developing both in vitro functional assays as well as in vivo assays to try to study the biology of T-lymphoblastic leukemia. And also, the power of the animal model is that we can treat them in a host to actually look at not only disease response, but also toxicity. So I want to talk a little bit about the functional assay, the in vitro assay that we used, and it was actually discovered not too long ago that, even though these are very aggressive leukemias, nobody can grow them in culture. Nobody can actually maintain them in vitro long enough to actually study meaningfully their biology. And so we actually had to develop an assay to maintain these cells. And in Toronto, there is a group led by J.C. Zoniga-Flicker, who actually, in studying T-cell development, transfected the Notch ligand DL1 onto a bone marrow stromal line OP9. And this OP9 DL1 stromal line has been very useful in studying the various stages of human T-cell, mirroring as well as human T-cell development. And its functional properties are largely unique, probably because of some of the cytokines it secretes, but also because it actually expresses in high density the ligand for the Notch receptor. So the Notch receptor family is a well-conserved family of cell surface receptors, which are highly expressed in many organs, and certainly in development. It's highly expressed during periods of organogenesis or development where cell fate is being decided. And so Notch actually regulates a lot of different stages of T-cell development that includes not only cell fate, but proliferation. So I'm just highlighting again Notch playing a very critical role in normal T-cell development, and when T-cells decide to be T-cells instead of B-cells, Notch also plays a very big role in expanding that progenitor pool. So certainly Notch is a very critical player in normal T-cell development, but even more so, it has been found that it's actually the most commonly mutated gene in human TALL. And so having a stromal cell line that express a ligand for this receptor clearly was our top pick for what would keep TAL cells alive in vitro. So what we did was use the co-culture assay to see if we can actually maintain these TAL cells in vitro, which, like I said, has never been done before. And we actually compared different combinations of cytokines in order to look at the growth over time. The other assay that we use is the in vivo model, and initially we were using the non-skid model with the help of an anti-CD122 antibody. The NK target, CD122, was definitely very helpful because if you just used non-skid, NK cell function that is retained in this TB deficient mouse still wasn't enough to encourage xenografts. The yield was still fairly low. What my mentor, Dr. Dick's lab, had found was that by using anti-CD122 antibody to eliminate NK cells, improved engraftment significantly. We also did our approach differently than other groups in trying to inject the leukemia cells directly into the niche. So we did intrafemoral, intra-bone marrow injections of the leukemia cells, and then waited for the cells to engraft. We were routinely irradiating the mice to make room in the bone marrow for the xenografts. But in fact, one of the, and I'll speak on this a little bit more, one of the concerns with respect to even low-dose radiation in this anti-skid model is that skid in fact encodes a DNA repair defect. And any form of ionizing radiation, no matter how low it is, does induce DNA damage and the potential for neoplasm development in these xenograft hosts. So when we wanted to assay the biology and study the biology of these human T-lymphoblastic leukemias, we would take one of our patient samples, we would inject a certain quantity, the range of which is usually 3 to 4 log folds, so that we can actually look at the frequency of the leukemia-initiating cell, and then we would harvest the organs of interest, and then we would look for readouts of the response or recover the cells for our other functional studies. And as you can see, depending on the particular leukemia, which is labeled by their patient de-identified codes at the bottom, each leukemia had a different efficiency with respect to engrafting in our NS122 model. Definitely compared to just intravenous injection, there was a big difference in terms of the efficiency of engraftment. And engraftment is not hard to tell. I mean, you can see very obviously when you're engrafted. And this was important because for our studies looking at treatment efficacy and treatment response, it was very important for us to be able to determine engraftment before we treat. And actually, most of the time, what we did in order to have a live host for treatment is we would just do bone marrow aspirates and look for engraftment. Now, for many of the samples, the NS122 model worked very well, especially for the patients who are, who have very aggressive disease. The leukemia will engraft very, very quickly. But there are some, and this is the interesting thing about the biology of TAL. We call TAL no matter what the biology is like. And so there are some patients whose counts are sky high, but they actually have very indolent disease, and you can do the leukapheresis, and you give them drugs, but it's a very slow progressing disease. And for those ones, some of them are, we have billions of cells that we can inject, and it still is very difficult for the cells to engraft. And so it was not always possible to model the patient's particular disease in the NS122. The other problem, as I mentioned, is that the immune deficiency is probably not severe enough, and even the anti-CD122 was not sufficient to overcome that, and that might be another limitation to engraftment. And even low-dose radiation was sufficient to cause the mouse's own endogenous leukemia or lymphoma to develop, because of course lymphoid progenitors are still present in the skin, and so they could actually die well before engraftment from a endogenous lymphoma. So the question was, what's next? Can we use something that makes a better model? And so most labs now that use xenografts for their work have moved on to the NSG model, and this is a way more efficient host for engraftment of human cells of any kind. This is actually from a paper that showed melanoma in subcutaneous implants even. So it is a much better host. Engraftment was much faster, efficiency was higher, and they were much, much less susceptible to endogenous lymphoid tumor development. So we employed that one. And you can see we had much better engraftment even on this low cell count injection doses. We were able to get engraftment, and even the leukemia samples that would never engraft, no matter how many cells we put in, we saw good engraftment in the NSG. So as I said, we did a lot of functional assays. We already published what we thought was an identified population within TAL that was responsible for maintaining and causing the high-risk TAL that's published a long time ago. But I think the power of this model is really to allow us to model the patient's disease and study it and look at what are the mechanisms driving non-response or disease relapse. So using this model, we can actually give the mice chemo or novel agents and see what the response is like. And so using the same approach, inject the patient sample into the mice and wait for them to engraft. We can then treat them with standard therapy wait until they recover, and then harvest the end organs for evidence of minimal residual disease. We can do clonotypic analyses. We can look at gene expression arrays. We can look at all of those to identify usually a clone or a few clones that manage to evade the chemotherapeutic treatment. So I just want to give you an example of how we use this model. So this particular sample was a patient who had a very good drug response, good outcome, and when we inject these leukemia cells into the mice, we have very high engraftment, 90% of the bone marrow is taken over by leukemia. We gifted the mice drugs, and you can see that the leukemia is almost undetectable a week later. The flow cytometry histogram at the bottom row shows you an immunophenotype that we were tracking with respect to the good versus bad TALLs, which in our publication we suspected was an immunophenotype associated with more commonly with drug resistance. And so in this good, low risk TALL, you get very good drug response, and the few cells that are remaining are the CD1A positive T-Lymphoblastic leukemia cells. But for those patients who didn't respond or relapsed quickly or had a bad outcome, we could also model that disease, and it would engraft actually quite successfully in these NSG mice, and you can give them all the drugs and they would mimic the human disease. They don't respond and they get sick very quickly. The interesting thing is that when you look at the immunophenotype profile of the cells that persist, the leukemia cells that persist, you'll see that in the patient sample, it was mostly CD1A positive. You would think that they should have a good outcome based on what we identify as the cancer-initiating cells. But over time, what you see is that progression from a largely CD1A positive to a CD1A negative population. And in fact, in the patient himself, the leukemia actually showed a similar picture of probably a clonal shift. So what we found was in the good versus bad leukemias, the TAL cells that express the immunophenotype with 1A high seem to respond by having all the 1A cells wiped out and no emergence of the 1A negative, whereas the ones that didn't do well, either the 1A cells showed up early or they basically were the emerging clone after treatment. And when we looked through clonal mapping using junctional sequences for these leukemias, what we found was that in the for the high-risk leukemias, low responders, that there is clearly a clonal population that dominates after treatment, and some of the other clonal subsets disappear. And this, again, is cancer biology. Even if you have a very low-frequency clone that is drug-resistant, it will emerge. And so you can see the 1A negative population is clearly the one that persists after treatment. And even though the patient's original leukemia had evidence of multiple clones, the clone that persists is the 1A negative one. So I just want to show you that the TL xenograft model is definitely a feasible, we think, relatively high fidelity model for looking at patient-specific drug response. And what we found, certainly in the 25 samples that we have been able to assay, is that the 1A negative population is clearly one that is of concern in terms of the immunophenotype of cells that exhibit drug resistance. And these are being retransferred into serial xenografts to further study them. And this is actually what actually most TL xenografters are doing, because this is actually a very efficient way to study the clonal evolution in a surrogate host ahead of the patient's own biology. Now, it's very interesting, at the time, nobody really was paying much attention to immunophenotyping and TAL being subdivided into low-risk, high-risk categories, because I mean, TAL was all being treated as high-risk anyway. But completely independent of what we had found, there were other groups that clearly identified TL subset that was high-risk and 1A negative. And this was classified as the early T progenitor TL, early T precursor TL. And this was identified because in places like St. Jude's and other large leukemia treatment centers, they were also asking the same question. How can we risk stratify our TL patients? What are the bad responders looking like, and what can we do to identify them in advance of giving them useless therapy and moving them on to rescue therapy? And what they have found was that in general, the more immature and less T-cell-like the immunophenotype, the greater the risk of non-response. And so that's why they call it the early T precursor leukemia, because usually these are CD4, CD8 negative and express CD5, CD7 high, maybe even CD34, which is a hematopoietic stem cell marker, and don't express any CD3 or T-cell receptor antigens. So when we identified the high-risk population being 1A negative, and then somebody, actually two other groups, both in the UK and St. Jude's, seeing this, we were very excited. And so again, you can see the immunophenotype profile of T cells. They express CD34, and then it disappears, and then 1A is a certain cell surface molecule that only gets expressed upon maturation. So the 1A negative population on the left-hand side is the most immature. So why is this 1A negative immunophenotype potentially bad? Well, when you're so immature that you're not really T-cell-like, you don't really have the same response to things that are toxic to T-cell progenitors. And so dexamethasone, we all know, is very toxic to double-positive T-cells. You know, you give a dose of dex and the thymus shrinks down to nothing. And also by targeting cell cycle activity, which most TL are associated with mutations of cell cycle regulators, that's great if you're a double positive T cell that's already made its way through T cell development. But if you're a very early progenitor and you're not even entering cell cycle yet, cell cycle inhibitors aren't going to do a whole lot. Certainly, the 1A negative could be a surrogate marker for some other biologically distinct population yet to be defined. And so, for example, in human TL amongst adults, it's a totally different spectrum of genes that are mutated. So, finally, to kind of bring this to full circle, certainly through collaborations with St. Jude's and UHN and other labs, the genetic profile of this early T precursor leukemia has been now exposed, and it explains a lot of the biology that distinguishes this from the garden variety good response TL. And so Charlie Mulligan has done a lot to characterize the genomic signature of many of the leukemias, and fortunately he was able to target this particular condition, TL, which is very hard because there are not a lot of biobanks that have sufficient samples. And clearly, the ETP-TL is a much more primitive leukemia than most TL. And in fact, the genetic signature of TL is actually more like AML, which is the myeloid leukemia. So you remember the leukemia the leukemias are identified based on their hematopoietic lineage. And in fact, the signature of ETP-TL is actually more like AML. And so what they found at St. Jude's is, if you have ETP-TL, don't get TL protocol, get AML protocol, and you'll get a much better response. And in fact, there's now a trial undergoing in our center, along with St. Jude's and others, to actually, validate that the treatment is matching the genetic signature. So in summary, I just want to show you the power of the xenograft, even though it's very painful sometimes and laborious and expensive, that it really does offer very important information on a biological condition that we would otherwise not get to study in the human host. And the modeling of the treatment to the disease in the xenograft is so important to give us preclinical information as well as clinical information. And I'll just quickly talk about some of the things that we've been working on with industry to, again, use this tool to screen new agents. And I don't want to spend too much on this because, of course, everybody hates seeing all the maps of the signaling pathways, but signaling pathways involved in cancer proliferation obviously are prime targets for delivering targeted treatment with the minimal toxicity, and talk about some of the other models that we can try to use to get this data. So again, what are the good agents that we haven't tried yet in TAL, and how can we use this model to give us useful information? So I already told you about some of the limitations of the xenograft model. So in collaboration with Graham Dallaire and the group in Dalhousie, we've actually been doing collaboration looking at both zebrafish xenografts versus mouse xenografts to model human TAL. Now, I grew up in the lab in a generation predating zebrafish, and so I learned about this really through my collaborations with Jim Whitlock at SickKids and the TAL treatment group at COM. And the fact is, zebrafish is a very commonly used tool now in immunology because of the easy turnover, the transparent skin, so you can label the cells, you can actually see exactly where the cells go. It's very easy to maintain them. You have a fish tank in the lab, you can grow your own food. It's very cheap compared to mice. And most importantly, the turnover time is so quick that you can get a much faster readout of response versus non-response. You can just throw the drug in the water and they get treated. So certainly in the mouse model, where you have to spend many, many days to actually get engraftment and treatment readout, the zebrafish actually is one of the ways where we may be able to get some high throughput response results for drug response. The problem is it's never been validated. It's a fish, and we don't know really how good it is for replicating human disease. So as I indicated, the beauty of the zebrafish is you can see right through them, and so if you GFP or otherwise fluorescently label your cells, you can see exactly where the cells go, and you treat them, and suddenly those green color cells disappear, and you know that there's response. Much easier than trying to do a bone marrow on a mouse. So one of the drug targets that we've looked at is obviously Notch. I already mentioned how Notch is a very key player in human TAL, and we have worked with Genentech on their anti-Notch treatment. But there are a variety of anti-Notch treatments, and I'll go through a little bit about why Notch is a hard target for treatment. So again, Notch is a family of transmembrane receptor proteins that are highly conserved, and the activity through Notch is mediated by the intracellular component, which is the transcriptional regulator. Once it regulates into the nucleus, it has downstream targets, which actually people don't know exactly what they are. That results in activation of cell cycle activity, its proliferation, or even harder to identify targets for cell lineage fate. So as I said, many cancers have had identified Notch mutations, and certainly TL is one of them. It's again the most common mutated gene in human TLL, but it's not only TL. Even B-cell malignancies like CLL have documented mutations of Notch. So Notch inhibition is not just an area of interest for TL or BL, and certainly breast cancer and other cancers as well. Notch receptor targeting. Most of that work has been done as phase 1 trials for solid tumors, but the fact is nobody is yet doing that for leukemias. And as you can see, the most common side effect are GI side effects, and that's because Notch4 is highly expressed in the gut, and so if you have a non-specific GSI, that's going to knock out every Notch receptor. The gut was going to be an easy target. So working with Jim Whitlock, we're hoping that the next generation GSIs are actually good, more selective Notch inhibitors that we can try, and certainly this is the power of using the xenograft because we can actually see what the toxicity is like as well as what the drug response is going to be like for leukemia. And again, the power of doing it in the xenograft models, we can also combine it with other drugs to see what combination profile looks like. We've used Anti-Nonch1 antibodies, which are supposed to be very highly selective and target the Notch1 receptor and none of the rest of the Notch receptors. We've used the antibody that's humanized, which has a basically longer half-life and less of a tachyphylaxis response. And it actually, the other benefit is that Notch1, when mutated, is often a truncated receptor and wouldn't be that useful to for other antibodies or GSIs where they don't target the, when they're targeting the expressed full-length Notch receptor. So the antibody that we used was actually quite good for targeting even the truncated Notch receptor. So again, using this xenograft model, we engrafted a cohort of NSG mice with leukemia, and the control cohort would get standard treatment or control antibody or the anti-Nonch1 receptor, and then we would wait until the treatment was done, and then we would recover the cells to look at the response. And as you can see in these two representative plots for two specific leukemias, there were definitely some leukemias that responded better, some that didn't respond so well, and that's because half the TLs have a Notch mutation, and the Notch mutated leukemias definitely responded better to anti-Nonch1 receptor treatment, whereas the non-mutated, the wild-type leukemias had a very marginal any response. But the interesting thing, and we were sort of beat to the punchline to this by Katrina Jameson's group in UC San Diego, is that it's not just that there was a decrease in the leukemia in the primary xenograft. When we recovered these cells into and injected them into the secondary mice so that we could follow the biology and activity of these leukemia cells over time, we found that the Notch1 treated and responsive leukemias, even if the initial cell counts didn't drop by a lot, by the time you serially transplant them into the next cohort of NSGs, there was no engraftment. And so the results of this serial xenograft experiment is to suggest that Notch inhibition may not be cytotoxic enough to wipe out all of the bulk of the leukemia, but its initiating potential is gone down. So the clinical correlate would be you don't see this massive sudden drop in the leukemia cells, but over time you will see it disappear, and for the Notch mutated leukemias, that may be sufficient to gradually make it disappear and never come back. And of course, when you look at the toxicity profile, we can weigh the mice, we can check how much they're eating, we can check their cell profile and make sure that they don't have bad cytotoxicity to their bone marrow compartment or their secondary hematopoietic organs. You can see that in fact this anti-Nonch1 antibody really does work because you do wipe out all the cells in the thymus and affect the T cell population. And the last approach to targeting Notch would be to target it from the ligand side. So anti-DLL4 antibodies have been around less time, and so there's not as much of that available, but it certainly is an interesting approach to targeting Notch because it also has anti-VEGF potential, and so you can target not only the cancer itself by preventing the delta-4 Notch interaction, but you can actually suppress the local environment's ability to support the cancer cells. So could this be the approach to supplant cancers? You know, instead of wiping out cancers, we should just change the microenvironment to make it hostile to cancer cells? I think that's actually a very intriguing approach because this may actually be an approach that allows for very localized therapy, especially for solid tumors, without the systemic toxicity of the typical chemo agents. So again, some of the other concerns with respect to Notch inhibition, you know, there are gut side effects, the angiogenesis issues, and actually, one of the concerns is that Notch inhibition may actually potentially cause some neoplastic changes as well on its own, because Notch is such a pivotal player in cell fate, proliferation, and so forth. There is a question of whether or not prolonged treatment of Notch inhibitors might actually be detrimental. So I have just a little bit of time left, and so I thought I would be remiss not to talk about some of the regenerative medicine things that might be of interest to this group, and certainly is something that's near and dear to my heart, which is looking at airway reconstruction. So do I have any questions, or should I save it to the end? Save it to the end?
Unless there's a burning, we'll see prepared ways.
Okay. So we have a team that works on airway reconstruction because these are very sick and complex patients, and I want to obviously thank the team who have supported the care of these very sick patients. So to be born with an airway obstruction is a very bad thing because you're supposed to breathe. You know, when you come out of mom, and if you don't, then it's very hard to stay alive. And certainly how sick you are with this condition depends on how much of your airway is affected. And it's classic. It's a biphasic stridor. And sometimes these kids are scraping by for a period of time, but as soon as they catch the flu that season, they're in ICU. And the earliest we've seen it is obviously in the newborn, but we haven't even seen it presenting as late as adolescents. And like I said, these are often complex patients. They often have other anomalies associated with their airway obstruction. We don't know why. We don't know why this happens. We don't understand foregut development sufficiently to understand why tracheal rings go from being C-shaped to complete rings and therefore narrow the airway. This is I'm talking about fixed airway narrowing and not airway compression from blood vessels or other things, which are more dynamic airway obstructions. So I'll focus on the fixed airway obstruction. That's airway obstruction from complete tracheal rings, and it's usually classified by the type, by the anatomy. So you can have type A, which is short segment, B, which is funnel shaped, and C, which is every tracheal ring is a complete tracheal ring. So this is a bad problem. So what can you do? You can do nothing, and you can just hope and wait for the child to grow to see if the cartilage will increase in size sufficiently for them to outgrow the limitation of the narrow airway. And certainly it is possible for them to outgrow it because we know the airway grows. It grows at a relatively fast rate, well, relative actually to their body surface area. However, this is only a feasible option if you have a very short segment that's involved, and if the caliber is not already so narrow that you're already obstructing your airway. Usually what happens is they need some sort of surgical intervention, and if it's a short segment like type A, then you can just resect up to 3 rings and hook the 2 ends together. The other option is you open up the narrow segment and try to patch something in so that you stent open the airway with something else. And Usually that's cartilage. Some other centers have used pericardial patch. We have chosen to use the slide tracheoplasty, which is basically autologous plasty of the trachea. There have been other people, including our ENT department, that have deployed intraluminal stents to try to stent open the airway after they balloon dilate, disrupt the tracheal ring. And sometimes you just can't do any better than just venting these patients. And so some of them will go for trach and on positive pressure ventilation for support. So this is just to show you what the cartilage patch or pericardial patch tracheoplasty looks like. It's usually just done to the chest. You get the airway out and you stick a piece of something in to increase the caliber. And the picture is very pretty and very simple, but this is what you're really looking at because the trachea is well behind the great vessels, and you often need to relax the retraction of it because the patient doesn't have the cardiac output anymore. And so in between the registerable blood pressure, you have to put that patch in. Some people have also tried a posterior tracheoplasty where basically you break the complete ring up posteriorly and stitch it to the esophagus to try to stent it open. This is not very common, and it also made the airway prone to granulation tissue because the esophagus is really not good airway tissue. So as I said, we do mostly slide tracheoplasties, which is really the geometry of making a tube that is like this go onto each other, so you double the radius of the lumen, which then to our squares the cross-sectional area of the trachea. It's the patient's own tissue, so there's no, not a very significant risk of granulation tissue, but it does significantly shorten the airway. And so very few centers actually deal with this because this is such a rare diagnosis and the patients are very sick. And so every center has had its own treatment of choice. We don't really know what's the best treatment. And these patients are so complex that, they may actually die from another diagnosis that they have rather than the airway. The other thing is, once you've done the tracheoplasty, you are maintaining the airway, cleaning the airway, bronching them, and doing all the things that need to be done while they're intubated and even after they're intubated, making sure that the airway stays open. And by staying open is, a 1 millimeter airway going to a 2 millimeter airway, and then you still have to do a lot of the patency maintenance care. So historically, this is a lethal condition, uniformly. Okay? So when we started seeing these patients in large cohorts and treating them, we were doing the slide tracheoplasty. We published our results. The patients are very young, very complex, and many had dying spells. Many were in the ICU with severe respiratory acidosis. Many of them were well until they had a coincident respiratory infection, and so they were intubated for their respiratory failure. And this is just a CT reconstruction of the airway. You can see that tube, that very narrow, that's the endotracheal tube. That's not the airway. The airway is actually so small that it's basically the endotracheal tube. So when we prepared these patients, it was basically out of desperation. And when we did the tracheoplasties, most of them were already intubated for quite some time, quite sick, and we used bypass in the majority of the patients because they're just too unstable. And interestingly, at the time of their repair, we always bronch them, and the bronch always shows no lumen, whereas the CT is much more optimistic. It shows some lumen. So this is what it looks like on bypass when we do the slide tracheoplasty. So you'll see this is actually the proximal half of the cut airway, and this is the distal half of the cut airway. And so you see that we've created a slit on the back and a slit in the front, and that's how we slide them back together. And as I said, that does shorten the length of the airway. There were a few patients that were well enough that we didn't have to manage them operatively, and as I said, there were some that were just so severely affected, so sick that they were palliated. And what was the outcome? They were sick and many died. And the ones that died were the ones that had a associated anomalies. There were newborns, and the patients who weren't neonates and had especially no cardiac anomalies, they actually had a much better survival. We had our share of complications, and the complications were not inconsequential. And so what we showed is, don't give up on these kids. They actually do have a chance, and especially if they're not neonates or significant cardiac anomalies, they actually may have a chance for survival. And when we compared our results with other centers that have reported their outcomes, we found that even other centers were seeing similar results, which is if you're a newborn and you have a cardiac anomaly, your risk of dying was very high. But if you were done on bypass, you're a big kid, you didn't have cardiac anomaly, it didn't matter whether you had a slide or a patch, your survival was good. So again, newborns, bad, cardiac anomalies, bad. So what's the future for these kids? I mean, if there are newborns and they're cardiac anomalies and they're going to die, is there nothing that we can do for them? So you may have seen recently a couple of publications looking at the potential for using decellularized allografts to reconstruct organs and transplant them. And certainly the airway was one of those organs. And what the process involves is you have to harvest a cadaveric graft, a cadaveric trachea. You have to decellularize it, put it into a vascular bed or some sort of bioreactor to repopulate the trachea with cells. You then put it in to a recipient. And in the initial process, it took up to 6 months to go through this. And this is actually out of the New England Journal report that instead of using a bioreactor, they just implanted it into the forearm of the patient, the decellularized allograft, and then they put the patient's buccal mucosa cells into the tracheal allograft to populate it with epithelial cells, and then you see it basically repopulate the graft over time, such that after 6 months they could actually put a piece of it back into this patient's trachea. And the nice thing is that you start with the decellularized graft and you put the patient's own cells in, so it's an auto transplant essentially, and you don't make immunosuppression. And one of the problems, especially with respect to immunosuppression, is if you're going to be doing it in small babies and kids, that's lifelong treatment. So the other obvious requirement is that it has to be a vascularized graft because cartilage will die very quickly, and so whether you do the host's own bioreactor or you have a bioreactor that is ex vivo, the ability to deliver sufficient vascularization or nutrients to the cartilage are significant limitations. And there are people out there doing all sorts of airway transplants in the rogue manner and not using protocols and not using vascularized grafts. And I think I was mentioning to Andrew yesterday that, Maccarini and, basically he's been banished from the UK anyway, and they really don't want him to randomly do these things without standardization. And so we've actually been looking at this in Toronto, and I have Siba Hiko's picture here because she actually was the grad student that had done most of this in Toronto, looking at the role of decellularized tracheal allografts in adults, and we're looking at it now for infants, and how you need to really study whether the epithelial surface is repopulated with the right mix of epithelial cells, because there's not just one type of epithelial cells. Developing the protocol to use the bioreactor to repopulate, to support the cartilage, because actually it's not enough just to have the scaffold in epithelial cells. You also need to have ways to keep the cartilage integrity so that your airway doesn't suddenly just collapse, because that's still airway obstruction. And so this is actually some of the studies that we're doing now to look at how to maintain the airway integrity from this cell-rise allograft repopulated with patients' cells. So in summary, this is a very quick overview of a rare condition, but it's a rare condition for which we have no good answers right now in terms of treatment. And obviously, if there is a potential to use regenerative medicine for this patient population, it may be the line of hope. Thank you very much.
We have time for some questions. Very exciting stuff. Yes, go ahead.
I just have a question about your TAL talk. Very interesting. I was wondering, since immunokines are intimately associated with maturation and development of T cells and adhesion of T cells and even Notch receptor expression, I was wondering, so you've immunotyped the responders and non-responders with respect to the T cells. I was wondering about the immune milieu, in other words, in the patients, if that's been looked at as far as patients that respond and don't respond.
Well, actually, looking at the niche has been a bit of a difficult target for any cancer, especially for the hematopoietic cancers where the bone marrow microenvironment is very difficult to study in patients. And certainly one of the things that, not our lab, but Mentor's lab, Dr. Dick's lab, has looked at is that particular question, which is the niche, the bone marrow microenvironment. And actually, one of his previous postdocs had actually done this work looking at the role of osteoclasts in the bone marrow, in the bone, actually playing a role in creating an environment in response to leukemia that actually allows for the expansion of AML. So that was particular. But you're right, there are many things that cancer cells have evolved to do is to, adapt to their environment or even modify their environment in order to promote their own growth. For TL, no, we haven't had the opportunity, but one of the things that we're trying to do is actually generate an ex vivo model that can actually look at the microenvironment requirements.
Hi, I have a very quick question. The leukemia work, and is there anybody trying to get the cells, leukemia cell from the patient to create a cell line and use peripheral stem cell? We're looking at the chemo or immune kind of response.
Yeah, actually, as I mentioned, TL, like many leukemias, they're actually very hard to grow in vitro. And for years, AML has been maintained in vitro using methylcellulose, just like bone marrow progenitors were maintained with methylcellulose. But for TL, there is no, other than on the OP90 and stroma, or other people now generated similar stromal lines, they really don't grow well in isolation. Now, that's not true for all TLs. Jerkat is like the world's oldest TL cell line, right? There's definitely TL cell lines that have been created, and there's some other ones. But to be honest, once, working with some collaborators, we've actually compared the genetic profile of cell lines versus primary tumors, and the fact is the genetic signature of a cell line is very different from an ex vivo tumor. But the sheer fact that you've adapted them to culture means that they've lost the need for cell-cell interactions. They don't grow well when they are dense. In fact, one of the things with some of these leukemias is they actually don't want to grow to density. Where some, because they actually need to grow to density, you can only maintain them at high cell numbers. So there are many features, once they adapt to culture, that really no longer mimic the primary tumor. And that's why actually most of our work has been not based on any cell lines, but really on the primary patient sample. And so you may use cell lines to, look at some target genes, but unfortunately, it may be very different from the primary disease.
Adult cell from the patient and then introduce a couple genes, keep them growing. induce the TS.
Yeah, and in fact, when you look at TL cell lines or any cancer cells that have been maintained in vitro long enough, one of the most common mutations that they acquire are cell cycle checkpoint genes, right? Because once they grow in vitro, they like to proliferate, so they select out the ones that already had, acquired the cell cycle regulators mutations.
Well, it'd be great to carry on this discussion, but it looks like we're past the hour, so thank you very much. And please come up and talk to us.
Sorry.
No, it's good.